Okay, so we are going to be talking about urine because that's one of the main functions of the kidneys, but the real reason we care about it is that urine is produced largely to control the solutes and the ions and stuff in the blood, and also to control blood volume. So we're going to be going through the kidneys kind of focusing on those things, as well as like the hormones that affect it, the different parts of the kidneys that are involved in different portions of like filtration and reabsorption. So we're going to be talking about filtration, secretion, reabsorption, and some of those words should be familiar from when we were talking about capillaries and the interstitial fluid. We're going to be talking about things like Starling forces again, stuff like that. But we're going to also be doing some other math. So we'll be talking about some things like filtered load, filtration rate, clearance, transport maximum, and renal thresholds. We'll also be talking about the events that occur during micturition, but that's basically urine production. Okay, so we're starting by trying to understand the solutes and the plasma, which remember is like the liquid part of the blood. And we're going to be thinking about how much of this plasma, how much blood flows through the kidneys at any given moment, and then how much of that is going to get filtered out as we adjust the solutes. Okay. So this is basically the function of the urinary system, right? We can also refer to it as renal exchange. And there are going to be different parts of the kidney that specialize for sort of different functions in this process of filtration, secretion, reabsorption. So as we go through the kidneys, and specifically kind of the microscopic anatomy of the kidneys, or the nephron is going to be like our unit of the kidney, we're going to be talking about what each part of that does, because this process is going to look a little different in different parts of our nephrons. Okay, so you're actually going to cover the gross anatomy of the kidney in anatomy lab. Do you guys all kind of understand? Sorry, I'm turning to face you, but of course that's not the camera. Did you guys click into the lab Blackboard and see how that works? No, not yet.
I wanted to see the schedule, but like I didn't like click into any of like the lectures or anything.
Okay. Well, you'll be doing this stuff in there. Yeah, basically that's going to be through recorded videos. Luis and Dr. Richards got access to the 3D VR headset things, and we're able, I think, to record a little bit of stuff in there. Doing anatomy not hands-on is kind of weird, but hopefully that'll be okay. So I think that those should be up really soon if you want to click in and check them out. Okay, that was last time. Okay, so when we're thinking about functions of the urinary system, so functions of the kidneys, we're thinking about regulating the ions in the plasma. And that's really important, right? Because we remember that, for example, our neurons, right, are really dependent on those concentrations of sodium and potassium. So it's really important to make sure that we have consistent levels of those in the blood, or else you're going to be messing up the membrane potentials of your nerves, your muscles, cells in the body that like actually have to do stuff by changing that difference in voltage across the membrane. So that's a really important function of the kidneys that you don't necessarily usually think about if you're just thinking about, you know, you pee, it's your kidney. They also regulate plasma volume. So when we were talking about arterial pressure and blood volume, blood pressure, we said that kind of long-term regulation is partially going to be dependent on the kidneys. And that's going to be happening through the processes that we're going to go through. So they're going to be regulating plasma volume, which is going to have effects on blood pressure because most of your blood is plasma, because most of your blood is basically water. And so if you retain more plasma or retain more water in that blood, you basically have more blood, which is going to raise your blood pressure. The urinary system is also important for regulating plasma osmolarity, which is kind of related to the ionic composition of the plasma, right? It just includes a couple more components, right? We're thinking about the insoluble solutes, so the things that can't cross membranes, and how that affects the osmotic pressure and the forces for filtration versus absorption, which are going to have effects within the kidney, but also effects in capillary systems elsewhere. We're also going to get to acid-base balance in the blood. So we're going to see that the urinary system, in combination with the respiratory system, is responsible for regulating pH in the body, which is going to be really important. And then the urinary system also removes metabolic waste products, which is usually the first thing people think about when they think about kidneys. It is important, but not necessarily actually the most important thing that the kidney does. Other things that the kidneys do is that they also kind of act as an endocrine organ. So they are going to produce some hormones. So when we talked about the development of red blood cells, we talked about how we need this hormone erythropoietin. So that's going to be secreted by the kidneys. And so is renin, which is going to be involved in basically the water balance in the body, basically. We'll get to that when we get to where water is flowing in and out of our nephrons. They're also important for activating some of our vitamins. So in order to actually use vitamin D3, it needs to be activated by the kidneys into something called calcitrol. And the kidneys can also basically make glucose from other things. So if you think way back to metabolism, we talked about gluconeogenesis, we talked about breaking down glucose. This is one of the places that that creation of glucose is happening. Okay. So when we're looking at the kidneys, if we're looking at the microscopic anatomy of a kidney, it looks something like this. So the basic unit of the kidney is something called a nephron. And I'm actually going to make you guys all draw me a nephron after class and label it. Some of the parts are labeled here. This is the best picture in the slide set, I think, that shows the structure. So I'm just going to kind of mention some of the stuff right now. So our nephron is this kind of swirly thing. So it's going to start with this structure called the glomerulus, which is basically a capillary bed. So we have an arteriole running into it, this capillary bed, and then an arteriole running out. And then we have this structure that basically catches fluid coming out of the blood in the glomerulus. It goes through a series of tubes. So it has these tubules. We got proximal ones, we got convoluted ones. So the proximal ones have a convoluted part and a straight part. So that's here. And then we go down into this big loop and we dip down into a different part of the kidney called the medulla. And we're dipping down. This is something called the loop of Henle. And this is where we're going to see a lot of things going in and out of the filtrate moving through this nephron. We're going to go back up. So we have an ascending part of our nephron. We're going to swirl around and eventually go into this sort of collecting duct over here. And this is what is going to link up into the gross anatomy of the kidney. So the part that you'll be able to see when we're looking at it in anatomy lab, that's eventually going to go into the ureters, come out the urethra. A thing to note about this is that, like, whenever we're talking about any of the parts of the nephron, we're going to have these really close associations between the blood flow and these tubules. So here we can see that we have blood coming out basically of our systemic system. We have blood going to this nephron. Afferent always means that it's going to something. And then we have our efferent arteriole, so it's leaving. This is the blood leaving. We can see how this blood is swirling around here. And the venous system is also swirling around this whole loop. So because we're talking about filtration and reabsorption and secretion, we're going to be talking about what solutes and what fluid is crossing from these tubules into the blood or back at any given point in this nephron. Okay. So when we're talking about renal exchange, what we're talking about is the stuff that's moving in and out of the nephron. So it's going to start here in this basically capillary bed that we call the glomerulus. So we have blood going to the beginning of our nephron through the afferent arteriole. Is that a question? No? Cool. Okay. So we have blood going through into the nephron through the afferent arteriole. And then we have these swirls of blood vessels, basically like capillaries, called the glomerulus. And then that blood leaves through the efferent arteriole. And I want you to note also that the efferent arteriole is kind of narrower than the afferent arteriole. And this is kind of important because it sort of backs up blood. So the blood is going to flow through the glomerulus relatively slowly so that we have time to filter out a lot of plasma. And the blood doesn't totally leave, and this is still arterial blood. So this is still oxygenated at this point. So don't get confused and think this is a vein because it's leaving. It's still oxygenated blood. So it's going to kind of swirl around, and then it's going to follow the rest of the nephron. So we're still going to have blood flow next to the tubules after we get past this first part, which we're going to call Bowman's capsule. Okay. So when we're looking at this diagram, the first place we have things moving in and out of the blood is in this area between the glomerulus and the structure called Bowman's capsule. So mainly what we're going to have happening here is filtration. So specifically, we're going to have basically everything but proteins coming out of the blood and moving into this tubule. So tons and tons of water, some solutes are going to be moving into our nephron here into Bowman's capsule. Then we're going to call the fluid here filtrate, and it's going to move through the rest of the nephron. And the whole nephron isn't drawn out here. That's why I was showing it to you on the previous page. So as the filtrate moves through the rest of the nephron, we're going to continue to move things in and out. So we have a ton of stuff here in the filtrate when we start out, but some of the things we've filtered out of the blood, we actually want to keep in the body. So those are going to get reabsorbed into these paratubular capillaries. So this blood flow that's running next to the tubules. And at this point, those paratubular capillaries may also secrete things back into the filtrate. So there's still really a close association between the blood flow and the tubule here. The filtrate is eventually going to get excreted, so it's eventually going to go out to our ureters. So yeah, get excreted from the body. So this is just a zoomed up version of this picture. We got blood flow going in, glomerulus, blood flow going out. Here we see where filtration is happening. So filtration happens first. We filter out a ton of plasma, plus other soluble solutes. Then we have reabsorption happening in our proximal tubules. We have secretion happening, so we have things moving back into the filtrate. And eventually whatever is still in that filtrate gets excreted. So our first big topic is going to be glomerular or filtration. So that's the start point where we have mostly plasma coming out of the blood that's moving through the glomerulus and into the structure that we call Bowman's capsule. So before on the previous slide in this picture, it shows Bowman's capsule just as this kind of cup, but the actual microscopic anatomy is kind of more complicated looking. So here what we're looking at is Bowman's capsule, and we can see that it also has cells kind of lying on top of the glomerulus, which is that sort of capillary bed. Okay. A ton of plasma actually moves out of the glomerulus at this point. So your glomerular filtration rate is 125 milliliters per minute. So that's how much plasma is moving out of the blood at this point, which if you add that up, it comes out to 180 liters per day, which is like a ton. That's way, way, way more blood than you have, right? So we can immediately tell that we're going to have to reabsorb some of that plasma and potentially some of the things dissolved in that plasma, because otherwise we're going to be out of blood in a couple minutes. Okay. So we can get so much plasma filtering out because of the structure of the cells that lie here around the glomerulus. So here we see a zoomed in part of the glomerulus, and here we see some of these cells that make up Bowman's capsule here. So the cells of the glomerulus are basically a capillary, and specifically they're basically a fenestrated capillary. So remember that a fenestrated capillary is where we have those endothelial cells kind of spaced out. So there's space between the cells for a lot of stuff to get through. We then have a basement membrane. So cells just sit on a basement membrane to hook them up together. And then on the other side, we have these cells that are part of Bowman's capsule. We're calling them podocytes. And that's because they have these little foot processes or pedicles that kind of spread out on top of the glomerulus. And they have slits between them. So we have these things called filtration slits, which are basically these gaps between the cells. And the size of those gaps, the size of those filtration slits can be controlled. So we can partially control what's coming in and out of the glomerulus by controlling, basically these gaps, by controlling these filtration slits. So here it is zoomed in. We see our basement membrane. So this part would be the inside of the capillary with those fenestrations. And here we see Bowman's space. I should say a thing about Bowman's capsule. So Bowman's capsule, if you think about like a bubble or a balloon and then you stick your fist into it, that's basically the shape of Bowman's capsule. So it has like a membrane here, basically, and then it continues and goes into the tubule. So when it says Bowman's space here, it's referring to the area like between the two layers of cells. So I'm going to scroll back again for a sec. So Bowman's space is this area here. So what we're looking at in this kind of cup area, so that would be like our podocytes. And then this layer of cells continues and starts to create the tubule afterwards. Any questions about the structural part right here yet? No? Okay. Well, here are more pictures anyway. So here is a tissue slide, actually kind of zooming in on this. So here we see what those filtration slits would look like. Here we see some of the foot processes of our podocytes. Here we see the basement membrane, and here we see the cells forming the capillary wall. So this fenestrated capillary. So what we've zoomed in on is just here. Okay, so controlling all of this, we basically have the same Starling forces as when we were talking about things moving between the capillary, the interstitial fluid, and out into actual tissue. But we're going to have specialized versions of this because we're talking very specifically about the glomerulus and Bowman's capsule and the fluids in there. So it's a similar setup, but the numbers are going to be different because we have slightly different pressures in the glomerulus and in the nephrons than when we were talking about just your normal capillary. So we're going to be tracking our hydrostatic pressures and our oncotic pressures, just like with capillaries. So we're going to have a hydrostatic pressure from the glomerulus. So our hydrostatic from the pressure from the glomerulus, or the glomerular capillary hydrostatic pressure, abbreviated PGC, is going to tend to push plasma out. So it's going to favor filtration. So filtration is moving from the blood into Bowman's capsule here. The other thing that favors filtration is the oncotic pressure of the capsule. So the oncotic pressure of the capsule here, here. This would be anything dissolved here in the fluid, in the filtrate in Bowman's capsule, would have a tendency to pull filtrate towards it. The actual normal value of the oncotic pressure of Bowman's capsule is about 0, because those proteins stay stuck in the glomerulus. So usually there isn't a pull in this direction. But if we had proteins leaking into the nephron for some reason, would usually be pathological, but it would affect this filtration rate. So we also have pressures in the opposite direction. As we saw on the previous slide, what is going to happen overall is filtration of plasma out, but that doesn't mean that there aren't pressures that oppose it. So we have the hydrostatic pressure of the filtrate in Bowman's capsule is going to push back the other way. And the glomerular capillary oncotic pressure is really the bigger thing, which is going to pull back towards the glomerulus. So here we can see the values for that. So our hydrostatic pressure here of Bowman's capsule is significantly lower than the hydrostatic pressure of the glomerular capillary. And we can see that the oncotic pressure of the glomerulus, so the glomerular capillary oncotic pressure, is much higher than the oncotic pressure of Bowman's capsule, because we still have all those proteins basically getting concentrated within the glomerulus.
Do you think we need to know those values? For the pressures?
Know the relative values. So like
Right.
know which one is higher, right? Like
Right.
it's
Okay.
important
Just
to know that like the
formula is
proteins
fine.
are dissolved here. So that's going to favor reabsorption back into the glomerulus, things like that.
Okay.
Yeah. But also, since I'm giving you the essay questions 24 hours ahead of time, you know. Okay. So here we're actually looking at the solutes dissolved here. So this goes through the forces favoring filtration and opposing filtration again. So we have our glomerular capillary hydrostatic pressure. Okay. So the hydrostatic pressure is pretty high because we have a relatively high like blood pressure in these capillaries. And this is because that efferent arteriole, scroll way back. So that efferent arteriole, remember I said it's kind of narrower than the afferent arteriole. So because it's narrower, it has higher resistance, which is basically acting as a blockage. So this is going to be higher, a higher pressure than the pressure in just your normal systemic capillaries. The oncotic pressure in Bowman's capsule is very low because we don't have these proteins moving in, so we don't really have solutes sucking more water towards it. So that would go with the Starling forces opposing filtration instead, because we have these proteins. I think that's the green here. Since the proteins are stuck in our glomerulus, they're going to pull water towards them because we want to water them down. But that's not a high enough force. So sorry, I'm doing these out of order. What I'm describing is the glomerular oncotic pressure. So it's pretty high, but it's not high enough to fully oppose the glomerular capillary hydrostatic pressure. And we also have the Bowman's capsule hydrostatic pressure opposing filtration. So like Bowman's capsule isn't empty, it does have filtrate in it. So there's also a pressure of that fluid back towards the glomerulus. Okay. This is written out even bigger with our arrows pointing the different directions. So we have our hydrostatic pressure from the glomerulus going this way. We have our hydrostatic pressure from Bowman's capsule going the opposite way. We have our oncotic pressure from Bowman's capsule moving us towards Bowman's capsule. We have our oncotic pressure from the glomerulus moving us towards the glomerulus. So we also want to talk about the rates that this happens. So going through the kidneys, so renal, just an adjective for kidney. So the renal plasma flow, so the amount of plasma that goes through the kidneys in any given minute is 625 milliliters. And 125 milliliters of that plasma is going to get sucked out into the Bowman's capsule every minute. So this is summing it up between all the nephrons in the kidney. So that's a lot more than when we were talking about the systemic capillaries. So in the systemic capillaries, our filtration pressure normally was only about 2 millimeters of mercury, whereas we now have a filtration pressure of 16 millimeters of mercury to move fluid into the nephron. So we only filtered out about 3 liters per day of plasma into tissue in our normal systemic capillaries. So that was how they were getting, right, you know, their oxygen, their nutrients through filtration in the systemic capillaries. That's much, much less fluid than is going through the kidneys. So we can also calculate something called the filtration fraction. So sometimes we're going to want to compare like how much of the fluid going through the kidneys is getting filtered out into the nephron relative to the amount of like blood flow. So our filtration fraction is just an expression of that ratio. So we take our glomerular filtration rate, which is the milliliters, those 125 milliliters of filtrate of plasma that we're moving into the nephron every minute. We compare that to the 625 milliliters of plasma that are going through the glomerulus at any given point. And we can express that as a fraction, so we can divide the 125 milliliters per minute of plasma being filtered to the 625 milliliters going through the glomerulus. So what this is to say, actually, maybe this will be a little more clear. Maybe you're not confused, but I think I can be more clear in what I just said. So as we're having blood move through the afferent arteriole into the glomerulus and out the efferent arteriole, not all the plasma is getting filtered into the nephron. So some of the plasma, some of that fluid continues in these arterioles. And so we still have plasma over here. So this is kind of telling us how much we've removed, because we also have some stuff bypassing the nephron, which is fine because it's going to go through the nephron again really soon because we're filtering out 180 liters every day. So that plasma will get filtered out at some point in the day. Now, using our understanding of glomerular filtration, we can also calculate how much of any given solute, so anything dissolved in the blood, gets filtered out. So we call that the filtered load. And basically all we have to do is take that filtration rate, so the amount of plasma that's getting filtered out of the blood, and multiply that by the concentration of that solute in plasma. So this is going to happen with any small molecules. So proteins are stuck in the glomerulus. They can't get through those fenestrated capillaries, but ions and stuff can. So small stuff can go through those fenestrations and those slits without any trouble. So we will sometimes want to know, say, how much glucose is getting filtered out into the urine. And you can think how that might be important, right? Like when you think about diabetes, that's one of the things they test, right? One of the signs of diabetes, I think, is like sweet smelling urine. Don't quote me on that. But we're going to want to be able to measure these things because it's going to tell us something about how our kidneys are functioning, and it can also tell us some stuff potentially about what's going on in the blood overall. So the quantity filtered out into the filtrate is what we're going to call our filtered load, and it's going to depend on that concentration in the blood plus the amount of plasma that's getting filtered out into the nephron. So if we're talking about the normal situation for glucose, it's mathematically easy, which is why it makes a great example. So we have 125 milliliters per minute is our glomerular filtration rate. So 125 milliliters of plasma are moving out of the blood and into our nephron. Our normal level of plasma glucose is 1 milligram per milliliter. We might express it as 100 milligrams per deciliter, same thing. So our filtered load for glucose is going to be that 125 milliliters per minute, which is our glomerular filtration rate, multiplied by the concentration of glucose, which conveniently is 1 milligram per milliliter. We just multiply that across, we get 125 milligrams of glucose filtered out per minute. Now this gets more complicated if you have a concentration that is not 1, but it's going to work the same way no matter what solute we're talking about. We'll do the equation the same. Okay, so we're filtering out 180 liters of fluid every day, but we're only excreting 1.5 liters of urine, which is less than 1% of that basically. So 99% of that plasma that we're filtering out is going to get reabsorbed back into the blood, which is really good because you wouldn't want your blood volume totally falling. Like you don't want to run out of blood. So that's really important that we have this reabsorption process. However, because we have so much fluid moving through our kidneys, even a small change to the glomerular filtration rate, so even a like tiny little increase in that ratio, right? Like if we raise this to, I don't know, 21 or something, is going to make a big difference because this is like a per minute rate, right? So over a whole day, it's going to make a huge change in the volume of fluid that gets filtered out and therefore excreted. So for that reason, our glomerular filtration rate is really highly regulated because it's a big problem if we suddenly have a ton more or a ton less fluid and therefore plasma moving out of our blood. Okay. One of the big things that affects filtration rate is the pressure in the arteries. So when we were looking back, we were saying that the pressure of the blood in the glomerulus, so that glomerular capillary hydrostatic pressure is one of our factors for filtration. So if we increase blood pressure, that is going to increase the filtration rate. But we actually have some kind of safeguards on this. So it plays a big role, but there are certain points at which your body is able to sort of control this even though you have pressure going on. So if you get really high blood pressure, it overcomes your body's ability to regulate this. But at lower pressures, even when you see an increase, sometimes we're going to be able to keep that glomerular filtration steady. Okay. So we're going to have both intrinsic and extrinsic ways that we can control the glomerular filtration rate. And these are going to be pretty similar to things we saw when we were talking about our cardiovascular system. Well, at least this first one is. So one of the main ways that we can control the rate of glomerular filtration is that we have myogenic regulation of the blood flow. So we've talked before about smooth muscle in the walls of arterioles. So can you guys see me actually? Oh, okay, you can. I've realized I've been talking with my hands this whole time. So, okay. So when you have this rise in blood pressure, so you have more blood coming through this afferent arteriole at higher pressure, it stretches the muscle in the wall of that afferent arteriole. And that causes the smooth muscle of that afferent arteriole to basically snap back. So the smooth muscle in arterioles contracts whenever it feels a stretch. So our afferent glomerular arteriole is here. So this smooth muscle in the wall would snap back if we have a rise in our mean arterial pressure. Another thing that counts as intrinsic regulation, because it's coming from the nephron itself, is something we call tubuloglomerular feedback. So this has to do with one of the sort of parts of the nephron. I'm gonna run us back to that nephron picture real quick. Okay. So when we look at, oh, this picture's, no, that picture's not fine because it's no blood flow. Okay, so this structure is our nephron. And right here, we have the glomerulus, which is what we've been talking about. So this is our glomerulus, and this is Bowman's capsule. And you can see that the nephron goes through here, comes down, but then it loops back up. So you can see here that we have part of our nephron that is basically crossing these capillaries again. So, sorry, these arterioles again. So here's our afferent arteriole, which is the blood flow going to the glomerulus. And we have our distal tubules going past that arteriole again. I'm going to skim back to that slide that we were on. Okay. So what we see here is that tubule coming back up. So all the stuff here has gone through most of the nephron already. And here it's touching the arterioles as it loops back, like right before it dumps into the collecting ducts and then into the ureters. Now, it has special cells in it, in the ascending limb of the nephron. So the ascending limb is the part just coming back up, that secrete something called paracrine factors. So paracrine factors are basically like our endocrine system, except for, so they're like hormones, but they just act really close. So they don't go into your bloodstream and affect the rest of the body. They're just kind of a local factor. So we have these paracrine factors that get released from these macula densa cells, and they can respond to increases in the flow of fluid going past them. So when we have more fluid moving through here, we have fluid moving faster, we have an increased flow. It can release these paracrine factors that are going to affect the blood flow in the arterioles. So if we have a lot of fluid coming up here through the end of our nephron, the macula densa will respond by secreting these paracrine factors, and they are going to cause the arterioles to contract. So that's going to cut off blood flow to the glomerulus, which is going to have downstream effects. So this is another way we can basically get the blood to slow down, increase resistance coming into the nephron so that we can decrease the glomerular filtration rate. Because if we have less blood moving into the glomerulus per minute, we're going to have less blood, less plasma filtered out per minute. We also have something called mesangial cells. And actually, people don't know that much about mesangial cells or what the factors are, or what they're doing. But basically they lie on top of the glomerulus in the Bowman's capsule as well. And they kind of act like, remember when we talked about the capillary sphincters, when we were talking about systemic capillaries? They kind of act like that. So they can squeeze down and increase the resistance in the glomerulus that way, which then backs up blood flow moving into it because it's basically like increasing the total peripheral resistance, right? Like if we increase the resistance, we're going to decrease the flow. So here is a zoomed in kind of picture of this in a number of different ways. So we're focused in here on the glomerulus. Here we have our afferent arteriole, which is actually the lower one. It's the lower one on all these slides because it just is the lower one. We got the efferent arteriole, so the one going away on top. And we can see that we have our nephron kind of looping back, okay? So if we look, let's look here. So we were just talking about this one, but here we have our afferent arteriole coming to the glomerulus. Here we have our efferent arteriole going away. And here we have our nephron looping back. If we look at the histology side, we can see kind of the same thing, except it's a little harder to look at because it's real cells. But here's Bowman's capsule. So here's actually Bowman's space. So you can see this space between our cells. And you can see this little kind of hole here. So this is actually our nephron. This is our ascending limb of the nephron. So this is like this part coming back up. So what we see here, like touching the glomerulus right next to Bowman's capsule, are our macula densa cells. Oh, they do have mesangial cells on here as well. So these mesangial cells are our third thing that could kind of control the resistance of the glomerulus, so they could contract and cut off blood flow a little bit that way. Yeah, we'll do this. Okay, so if we see a rise in blood pressure, we have ways we can control that. So a rise in the mean arterial blood pressure does create a rise in the pressure in the afferent arteriole. We're gonna respond to that by sensing the stretch of the arteriole. So that smooth muscle is then going to constrict, which increases the resistance, which is going to decrease the glomerular capillary pressure. We'll go through the rest of this on Wednesday, because I think our time is up, if I'm right. In order to keep us all on track, I realize that I've been talking a lot about the nephron, but we kind of skipped the anatomy slides because we basically missed a week of class and had to cut a bunch of stuff. So for Wednesday, I would like you to define and draw a nephron. So there's an example in your textbook, so it shouldn't be that hard. I've put an assignments folder up on Blackboard, and there's like a Google form in it. So if you take a picture of your definition and your nephron and upload it there, I will see it. We're going to do these sort of like practice writing assignments regularly. At the end of the semester, I'll pick one at random, see who did it, and if you did it, you'll get 5 extra credit points for the class. Okay. Questions at this point? No?
No.
Well, I'm here. Let me know. But that's it for today.
All right, thank you.
Thank you.
Stay safe.
You
Yes,
too.
Good luck with
Bye.
all that.